Mendelian genetics, named after the pioneering scientist Gregor Mendel, is the cornerstone of classical genetics and provides the fundamental principles of heredity. Mendel's work with pea plants in the 19th century laid the groundwork for our understanding of how traits are inherited from one generation to the next. His principles remain central to genetics, influencing fields ranging from medicine to agriculture. Here's an overview of Mendelian genetics and its key concepts. Gregor Mendel, an Austrian monk, conducted experiments with garden pea plants, Pisum sativum, between 1856 and 1863. He meticulously observed the inheritance of traits, such as flower color, seed shape, and pod color. Mendel's experiments were groundbreaking because he used statistical analysis to deduce the underlying patterns of inheritance, leading to the formulation of several key principles. Mendel's first principle, also known as the law of segregation, states that each individual possesses two alleles for each trait, one inherited from each parent. These alleles segregate or separate during gamete formation, so each gamete carries only one allele for each trait. Upon fertilization, the offspring receive one allele from each parent, restoring the pair. The law of independent assortment states that genes for different traits are inherited independently of each other. For example, the inheritance of seed shape is independent of the inheritance of seed color. This principle applies to genes located on different chromosomes or those far apart on the same chromosome. Mendel observed that some alleles are dominant while others are recessive. A dominant allele masks the expression of a recessive allele in heterozygous individuals. For instance, in Mendel's pea plants, the allele for round seeds, R, is dominant over the allele for wrinkled seeds, R. Thus, only plants with two recessive alleles exhibit wrinkled seeds. Mendel's experiments led to the development of Punnett squares, a tool used to predict the genotypic and phenotypic ratios of offspring from genetic crosses. By crossing plants with different trait combinations, Mendel observed predictable patterns of inheritance, which he described using ratios such as 3:1, dominant to recessive, in monohybrid crosses, and 9:3:1 in dihybrid crosses. While Mendelian genetics provides a foundation, modern genetics has expanded upon Mendel's principles. For example, concepts such as incomplete dominance, codominance, and chased polygenic inheritance illustrate that not all traits follow simple Mendelian patterns. Incomplete dominance occurs when neither allele is completely dominant, resulting in an intermediate phenotype. Codominance, on the other hand, involves the simultaneous expression of both alleles, as seen in blood type AB. Furthermore, genetic linkage, when genes are located close together on the same chromosome, can affect independent assortment. Advances in molecular genetics and genomics have provided deeper insights into gene interactions and the complexity of inheritance. Mendelian genetics has profound implications in various fields. In medicine, it aids in understanding genetic disorders and inheritance patterns, enabling genetic counseling and disease prediction. In agriculture, principles of Mendelian genetics guide plant and animal breeding programs to enhance desirable traits. In summary, Mendelian genetics forms the bedrock of genetic science by explaining the principles of heredity through systematic experimentation and observation. Mendel's principles of segregation, independent assortment, and dominance provide a framework for understanding genetic inheritance, while modern advancements continue to build upon his foundational work.